Good morning, students. Up till now, in the lesson Life Processes in Living Organisms Part 1, we have seen um, which are the various important life processes. Then we have seen glycolysis, we have seen TCA, that is tricarboxylic acid cycle, then the ETS, that is the electron transfer chain reaction. We have also seen Krebs cycle, how anaerobic respiration takes place, or how energy production in microorganisms through anaerobic respiration takes place, then energy from different food components. These are the things that we have seen up till last week. Now this week we move on to a very important and an essential life process, that is cell division. Page number 17. Cell division is an essential life process. Here, the first question that they have asked is, what happens to the cells of injured cells? So, when a cell gets injured, what happens? The cells and the tissues, they are not able to perform the regular function once they get injured. At the site of injury, the blood capillaries rupture and the area gets inflamed or it gets swollen. Then second, whether new cells are formed during healing of the wound. Yes, as the wound heals, there is scab developed, or the cells surrounding the wound start dividing rapidly, and the cells lost in the injury are restored back. Third question, do the plants get injured when we pluck the flowers? How are those wounds healed? Answer is yes, plants get injured when we pluck the flowers. And how are these wounds healed? New cells are formed, and in this way the wound is being healed. Fourth question, how does the growth of any living organism occur? Does the number of cells in their body increase? If yes, how? Any living organism grows due to the increase in the number of cells in their body. The cells divide regularly and add new cells which are essential for growth. The cell division is thus necessary for growth of the body and also for the regeneration and repairing of the lost tissues. So that is the answer for the fourth one. Fifth question, how the new individual of a species is formed from the existing one of the same species? The new, as you all know, the new species or the individual is formed by the process of reproduction. Now let's move on. Cell division is one of the very important properties of cells and living organisms. Due to this property, a new organism is formed. A multicellular organism grows up and emaciated body can be restored. Now, what is the meaning of emaciated? Emaciated means extreme weight loss or unnatural thinness due to the loss of subcutaneous or the adipose tissue which is present beneath the skin. That is the meaning of emaciated. In our regular language, we can say extreme weight loss or very, very thin. Right? Let's move on to types of cell divisions. There are two types of cell divisions as mitosis and meiosis. Mitosis occurs in somatic cells and stem cells of the body, whereas meiosis occurs in germ cells. So, this mitosis, which is one of the cell division, it occurs in somatic cell. What are somatic cells? Somatic cells are any cell other than a reproductive cell. And what are stem cells? So the stem cells are a mass of undifferentiated cell which is capable of giving rise to more cells of same type. That is the meaning of stem cell. Whereas meiosis occurs in germ cells. Germ cells are reproductive cells. Before study of cell division, we should know the structural organization of the cell that we have studied earlier. Each cell, as you know, is made up of nucleus. Apart from nucleus, it also contains many other cell organelles like mitochondria, then Golgi bodies, vacuoles, etc. Let us study the cell division with the help of this information. So, page number 18. Before any type of cell division, the cell doubles up its chromosome number present in its nucleus. So, here it is given that before the cell division, before any type of cell division, whether it is mitosis or meiosis, the cell doubles up its chromosome number which is present in the nucleus. Now here it is given, that is, if chromosome number is 2n, it is doubled up to 4n. Now here I would like to make a clarification. Chromosome number does not change from 2n to 4n as it is given. Under normal conditions, such change never happens. The chromosome number never doubles. During cell division, initially the centromere of the chromosome does not divide, only chromatid arms divide. Getting it? During cell division, initially the centromere of the chromosomes does not divide, but what divides? Only chromatid arms divide. The division of the chromatid arms takes place in anaphase. Thus, chromosome number 2n remains 2n. It never becomes 4n during mitosis. That is a clarification which I wanted to make. Can you recall what is the shape of the chromosome? See figure 2.5. The shape of the chromosome, as you can see, it is dumbbell shaped. Give its names in the figure. So the first is P arm, then is centromere, then the thread-like structure which is present in a bunch, it is called as Q arm, and the last one, that is a loop-like structure, it is the DNA. A pair of each type of chromosome is present in 2n condition, where a single chromosome of each type is present in n or haploid condition, and their structure is like the one shown in the figure given beside, which we have just now seen. Now let us move on to mitosis. Somatic cells, that means the cells other than reproductive cells, and the stem cells, that is a group of cells which is undifferentiated, divide by mitosis. Mitosis is completed through two main steps. These two steps are: first, karyokinesis, which means division of the nucleus, and cytokinesis, which means division of the cytoplasm. Karyokinesis, which is the first part, that is the nuclear division, is completed through 4 steps. Which are these 4 steps? These 4 steps are prophase, first, second, metaphase, third, anaphase, and fourth, telophase. Let us see each of this phase in short. As you can see in the picture beside, in prophase, condensation of the basically thin thread-like chromosome starts. Due to this, they become short and thick, and they start to appear along with their pairs of sister chromatids. Centrioles duplicate, and each centriole moves to the opposite poles of the cells. One most important thing which we have to remember is that nuclear membrane and nucleolus starts to disappear. You can see in the diagram, I have shown you 2 diagrams. One is early prophase and the next one is late prophase. So in prophase, the points which you can remember are as follows. First, Prophase is very important, and so we can say in prophase, condensation of thin thread-like chromosomes takes place. That means the chromosomes become short and thick. Formation of sister chromatids begin. Duplication of centrioles and movement of each centriole to opposite poles of the cells takes place. And last thing is nuclear membrane and nucleolus start to disappear. Second phase is the metaphase. Nuclear membrane completely disappears in metaphase. So you can see that there is no nuclear membrane in metaphase. Chromosomes complete their condensation and become clearly visible along with their sister chromatids. Then next is all chromosomes are arranged parallel to the equatorial plane, that is the central plane of the cell. Special type of flexible protein fibers, which is called as spindle fibers, are formed between the centromere of each chromosome and both centrioles. Now this we can sum up in 4 points as: in metaphase, completion of condensation and chromosomes takes place. Then second point, chromosomes are clearly visible along with their sister chromatids. Third, chromosomes are arranged parallel to the equatorial plane of the cell. Formation of spindle fibers between centromere of each chromosome and both centrioles can be seen. And last, nuclear membrane completely disappears. In prophase, what it is? In prophase, nuclear membrane and nucleolus, they start to disappear, whereas in metaphase they totally disappear. Third, that is anaphase. In anaphase, centromere split and thereby sister chromatids of each chromosome separate, and they are pulled apart in opposite direction with the help of spindle fibers. Separated sister chromatids, they are called as daughter chromosomes. Chromosomes being pulled appear like bunch of bananas. In this way, each set of chromosome reach at two opposite poles of the cell. This is what is given in anaphase. If I try to tell you point wise, it is: first, in anaphase, formation of daughter chromosomes which appear like bunch of bananas is seen. They are then pulled away from each other. Second point, splitting of centromeres with separation of sister chromatids of each chromosome takes place. Their separation begins. Movement in the opposite direction takes place with the help of spindle fibers. Third point, each set of chromosome reach at two opposite poles of the cell. And last point, separated daughter chromosomes can be seen. Now the last phase of karyokinesis, that is telophase. Telophase, the chromosomes which have reached at opposite poles of the cell now start to decondense, due to which they again become thread-like thin and invisible. Nuclear membrane is formed around each set of chromosomes reached at the poles. Thus, two daughter nuclei are formed in a cell. Nucleolus also appears in each daughter nucleus. Spindle fibers completely disappears. So if I am to sum up with telophase, we can say In telophase, chromosomes reach at opposite poles of the cell and their decondensation starts. Second point, chromosomes become thread-like thin and start disappearing. Third, spindle fibers completely disappear. And last point, two daughter nuclei form in a cell and reappearance of nuclear membrane and nucleolus. You can say that telophase is exactly opposite to that of prophase, if you want to remember in a proper way. So this is karyokinesis. In this way, after karyokinesis, cytokinesis begins. Now what is cytokinesis? Cytokinesis is formed from two words, cyto and kinesis. Cyto means cytoplasm, breaking up of cytoplasm. The cytoplasm divides by cytokinesis and new cells are formed which are called as daughter cells. In this process, a notch means a groove, is formed at the equatorial plane of the cell, which deepens gradually, means it starts becoming more and more deep, and thereby two new cells are formed. However, in case of plant cells, instead of a notch, a plate is formed exactly along the midline of the cell and the cytokinesis is completed. What is the significance of mitosis? Mitosis is essential for growth of the body. Secondly, it is necessary for restoration of emaciated body, wound healing, formation of blood cells, etc. So that is for mitosis.